In this video, we are going to talk about CRISPR-Cas9 mediated genome engineering in Bicyclus anynana butterflies. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR were first identified in E. coli as identical short palindromic repeats of DNA. It was discovered that when viruses attack, bacterial cellular machinery produces Cas protein complex, which along with the information from spacer DNA cleaves and destroys viral genetic information. In Streptococcus pyogenes, only one Cas protein called Cas9 is needed, which along with CRRNA and tracer RNA can cleave any double-stranded DNA. In our lab using CRISPR-Cas9, we have successfully knocked out limb and eye spot gene distilless, wing selector gene Apterus A, and genes associated with melanin pigmentation. In this video, we are going to describe the protocol using the gene engrail, which is expressed in segments of embryo, posterior compartment of larval wings, in eye spot center, and the golden ring surrounding it. The major experimental stages includes design and verification of sgRNA, injection of sgRNA Cas9 mixture, rearing hashed individuals, screening and genotyping mutant individuals, designing primers. Type libbase.org in web browser, select ensemble.libbase.org, select Bicyclus anynana. In the screen box, type the name of the gene, select the transcript and select the sequence of interest. Paste the sequence in a Word file. Now type CRISPR direct in web browser. Click on crispr.dbcls.jp. Paste the DNA sequence and in the specificity checklist, select Postman butterfly. Click on design, choose the best guide sequence and paste it in the template. To design primer for amplifying DNA region of interest, go to Primer 3+ and paste the sequence in the box provided. Change the settings as shown in the screen and choose the best set of primer. Synthesis of sgDNA. To prepare sgDNA, in a 1.5 ml microcentrifuge tube, add 3 microliter of Q5 high-fidelity DNA polymerase, 60 microliter of Q5 polymerase buffer 10x, 6 microliter of DNTP, 9 microliter of forward primer, 9 microliter of reverse primer, and 213 microliter of molecular grade water. Vortex the mixture and aliquot in 3 200 microliter tubes. Set up PCR reaction with settings as shown on the screen. Run the PCR reactions in 1% agarose gel. Image the gel using a gel dock system. Purification of sgDNA. To purify the PCR reaction, transfer the completed reaction volume to a 1.5 ml microcentrifuge tube and add an equal volume of binding buffer. Vortex the mixture for 5 seconds. Transfer the mixture to a purification column and centrifuge at 13,000 rpm for 30 seconds. Add 500 microliter of wash buffer and centrifuge at 13,000 rpm for 30 seconds. Discard the flow through and repeat the step one more time. Spin the moist column for 1 additional minute at 13,000 rpm. Transfer the column to your new tube, add 20 microliter of elution buffer and centrifuge at 13,000 rpm for 1 minute. In vitro transcription to prepare sgRNA. For the synthesis of sgRNA, add 2 microliter of T7 10x buffer, 2 microliter of T7 RNA polymerase, NTP mix 2 microliter each, sgDNA 2 microliter, ribolog 0.5 microliter, and molecular grade water 5.5 microliter. Incubate the mixture at 37 degrees Celsius. Leave the tube for 16 hours. Take the tube out, spin the mixture and treat with DNase. Run the reaction in 1% agarose gel. Amplification of DNA fragment of interest. In a 1.5 ml microcentrifuge tube, add 62.5 microliter 2x PCR bio TAC mix red, 5 microliter forward primer, 5 microliter reverse primer, 5 microliter template DNA, and 47.5 microliter molecular grade water. Vortex the mixture. Aliquot the mixture in 5 200 microliter tubes. Set up PCR reaction with settings as shown on the screen. Run the PCR reaction in 1% agarose gel. Verification of sgRNA via in vitro cleavage assay. In a 1.5 ml microcentrifuge tube, add 1 microliter Cas9 buffer 10x, 0.1 microliter Cas9 protein, 0.5 microliter sgRNA, and 8.4 microliter molecular grade water. Incubate the sample at 37 degrees Celsius and leave it for 10 minutes. Add 200 nanogram of the amplified DNA. Incubate the sample at 37 degrees Celsius and leave it for 1 hour. Run the reaction in 1% agarose gel. Embryo collection. Place few corn leaves inside butterfly cages and leave them for 30 minutes to 1 hour. Remove the leaves. Collect the embryos in a paper cup. Needle preparation. Set the conditions on the needle puller as shown on the screen. Place the needle in between the holders and start the hit pull cycle. Preparation of sgRNA Cas9 mixture. In a 1.5 ml microcentrifuge tube, add 1 microliter Cas9 buffer 10x, 1 microliter Cas9 protein, 5 microliter sgRNA, and 3 microliter molecular grade water. Incubate the sample at 37 degrees Celsius for 10 minutes. Add 0.5 microliter non-toxic food dye. Vortex the mixture. Preparation for injection. Prepare a Petri plate with thin stripes of double-sided tape attached to the bottom of the plate. Using a paintbrush, carefully arrange the embryos on the double-sided tape. Pipette 3 microliter of the sgRNA Cas9 mix using a 20 microliter tip and transfer the content to the needle by filling it from the back. Break the tip of the needle by gently pressing against the side of a petri dish under a dissecting microscope. Injection of sgRNA Cas9 mixture into embryos. Carefully inject a small volume of sgRNA Cas9 mixture into the embryos. Place a wet cotton ball inside the Petri plate. Embryos will take 3 to 4 days to hatch. Rearing hatchlings and screening for mutants. Transfer the larvae into a paper cup with 1 young corn leaf. Keep note of any changes in the phenotype and image the abnormal individuals. T7 endonuclease assay and genotyping of mutants. Isolate genomic DNA from mutant tissue. Amplification of DNA fragment of interest for T7 assay. In a 1.5 ml microcentrifuge tube, add 62.5 microliter 2x PCR Bio Tac Mix Red, 5 microliter forward primer, 5 microliter reverse primer, 5 microliter template DNA, and 47.5 microliter molecular grade water. Vortex the mixture. Aliquot the mixture in 5 200 microliter tubes. Set up PCR reaction with settings as shown on the screen. Run the PCR reactions in 1% agarose gel. Purify the DNA as shown before. T7 endonuclease assay on amplified DNA. Prepare 2 200 microliter PCR tubes. In each tube, add 1 microliter of amplified DNA, 2 microliter of 10x NEB buffer 2, and 17 microliter of molecular grade water. Set up hybridization reactions with settings as shown on the screen. Add 1 microliter T7 endonuclease in one tube. Incubate both the tubes at 37 degrees Celsius for 15 minutes. Run the PCR reactions in 1% agarose gel. Sequence the DNA fragments. Analyzing the sequencing results and determination of indel sites. Align the sequences along with the original DNA sequence using CLUSTAL W. Look for the indels at the expected site.